DNA Replication and Angelman Syndrome by Evelyn Molina. What is DNA replication? DNA replication is the process by which DNA makes a copy of itself. DNA contains the information needed to carry out cellular activity. It is a genetic material. When cells make a complete and exact copy of DNA to pass along to the next cell. In this documentary, I will be discussing the DNA replication and why it is important to know the process of how our cells replicate, and how the process of DNA replication can lead to many syndromes, especially Angelman syndrome. So let's begin. Well, this process starts with an egg being fertilized by a sperm. During the fertilization, these gametes fuse to form a single cell called diploid zygote, meaning it has two complete sets of chromosomes, one from each parent. And this process is defined through mitosis, which creates an embryo, a fetus, and eventually a human being. That human will have trillions of cells, and we have to make sure each one of those cells has the same exact DNA that was found in the original cell. This is accomplished through DNA replication. Part of the process of sexual reproduction is the ability of the cells to make identical copy of their DNA before cell division occurs. This specifically occurs on the cell cycle or cell division, which is a series of events that take place in the cell leading to its division and replication. The cell cycle has three checkpoints: cell growth checkpoints, DNA synthesis checkpoints, and mitosis checkpoints. It is regulated by cycling and CDK. It has different levels of cycling and that observe at different cell cycles, and also can be paused by the action of p53. The G1 phase is where eukaryotic cells grow. The S phase, where the replication of DNA in eukaryotic cells occurs. G2 phase is where it continues to grow. The whole process is called interphase. And then we have mitosis, which is the division of genetic material in eukaryotic cells, where it goes through prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. During S phase, we have to make sure that all DNA was copied. Mitosis is not found in prokaryotes. They go through a process called binary fission that occurs when a cell divides into two or more parts, and regeneration of these parts occur into separate cells. Prokaryotic cells that reproduce the binary fission are part of the process of DNA replication. This is a simple process where one circular molecule of DNA is hooked up to the membrane and then elongates the membrane in between the attachment points. On the top of the binary fission on cell 1, you can see one circular molecule of DNA and how DNA bacterium has replicated. As you go under the cell 2, the DNA is pulled to the separate poles of the bacterium as it increases size to prepare for splitting. On cell 3, the growth of a new cell wall begins to separate the bacterium, and this process consists of adding more and more membranes in between the two areas of attachment, and slowly is pinching between, and the process is done. In between cell 4, then we can see that the new cell wall was fully developed, resulting in a complete split of the bacterium. On cell 5, the process is complete by having a new daughter cell that have tightly coiled DNA, ribosomes, and plasmid. The structure of DNA was discovered in April 1953 by biologists James Watson and physicist Francis Crick. They solved the puzzle that several other groups were trying to solve at the same time. They knew some important facts about DNA. For instance, the science knew that DNA was composed of 4 bases: adenine, cytosine, guanine, and thymine. The testing of the theory was accomplished by American molecular biologists Matthew Meselson and Franklin Stoll in 1958, known as the Meselson-Stoll experiment. 3 new theories of how DNA replicates came out of their work: the semi-conservative, conservative, and dispersive. The semi-conservative replication produced 2 copies that each contain one of the original strands and 1 new strand. The conservative replication leaves the original template DNA strands together in a double helix and will produce a copy composed of 2 new strands standing all of the new base pairs. The dispersive replication will produce 2 copies of DNA, either both original strands or both new strands. Watson and Crick believe on the semi-conservative. They believe that DNA splits in half and then you copy a new strand on each side, but there are other scientists who believe on the conservative theory that the first DNA remains intact, while others believe that the combination of the semi-conservative and conservative. So those who believe on the dispersive theory, they focus on the histone protein. And all this was figured out through the Mendelssohn cell experiment. And they used two types of nitrogen, nitrogen 14 and nitrogen 15. The semi-conservative replication results in a double-strand DNA with one strand of nitrogen 15 DNA and other of nitrogen 14 DNA. The results of the experiments were consistent with the semi-conservative replication hypothesis. Semi-conservative replication can go into greater detail. The DNA in the top is started with one strand and end up with two strands, which are identical to the first strand, and each of the strands in the bottom contains half of the original DNA, and this is what is called semi-conservative by nature. Before we continue talking about the process of DNA replication, let's remember DNA and the parts of DNA. Remember, DNA has three parts: sugar, which is the deoxyribose sugar, nitrogen base attached to that, and the phosphate group. And these are the three groups to every nucleotide. As you can see, there are three groups in the antiparallel. And it's called antiparallel because two strands of DNA are running in a positive direction, chemically running in either direction. The way that we can tell is which direction is going, we do that based on the sugar. If you look at the orange sugar, that will have 5' sugar. So if you look down straight, that will run down to the 3' and all the way up to the 5' end. And if you look to the other side, you will see that it's running in the positive direction from the 3' end to the 5' end. And that is very important because when we look how DNA and how it copies itself, DNA replication, there are tons of enzymes that help out. Basically, DNA is a double helix, and there is an enzyme called helicase that unwinds the DNA, and this is how we go from a double helix to a single strand of DNA on each side. These strands are held in place by those enzymes called single-strand binding proteins, and what they do is basically they hold them in place. The big and important enzyme in this process is called DNA polymerase, which raises the DNAs and add new nucleotides to the other side of DNA. Remember that we can only add new nucleotide to the 3' end. We cannot add it to the 5'. The bottom of the picture is called leading strand, which means it flows perfectly. The top of the picture is called the lagging strand because it tends to lag to the other side. RNA primer is added first, then DNA polymerase can go through that direction. But once there is a space in between that cannot connect, a DNA ligase is used. And what is this basically does is clean these junctions, and then the purpose of the process is to message what it was found in DNA, and it's copied to a new strand of DNA on either side. And this is what is called perfect DNA replication. Now that we know the process of DNA replication, let's see how this process can lead to many syndromes in our society, and one of them being Angelman syndrome. Angelman syndrome is a genetic disorder. It's most often caused by problems in the gene located on chromosome 15 called the ubiquitin protein ligase, E3A, UBE3A gene. As we know, genes are segments of DNA that provide the blueprints for all your characteristics. You receive genes which occurs in pairs from your parents. One copy comes from mom, the other copy comes from dad. Angelman syndrome is caused during DNA replication with chromosome 15q11/q13, a region subject to genomic imprinting. This means that the cells use information from both the maternal copy and the paternal copy of each gene pair. But in a small number of genes, only one copy of the gene pair is active. The activity of each gene copy depends on whether it was passed from the mother or from the father. This parent-specific gene activity is called imprinting, and these genes, when the copy that is usually active is missing or defective, and it causes problems in functions and characteristics controlled by the gene. Normally, only the maternal copy of the UB3A gene is active in the brain, caused by what is known as genomic imprinting. Most cases of Angelman syndrome occurs in parts of maternal chromosome 15, which contains this gene, is missing or damaged. In a small number of cases, Angelman syndrome is caused when two paternal copies of gene are inherited, instead of one paternal and one maternal copy, which are paternal uniparental disomy. Most babies with this syndrome do not show signs or symptoms of the disorder when they were born. Angelman syndrome is rare, and the symptoms become noted at 6 to 12 months with development delays, feeding difficulties, lack or minimal speech, inability to walk, move or balance, trembling movement of arms and legs, frequent smile, and they're so happy all the time. People who have Angelman syndrome may also have other signs and symptoms. Some of them are developed between 2 and 3 years of age. They get seizures, stiff or jerky movements, small head size with flatness in the back of the head, which is called microbrachycephaly, using crossing the eyes, light pigmentation in the hair of the eyes, sleep disorders. Angelman syndrome affects an estimate of 1 to 20,000, and Dr. Harry Angelman is one of the ones who found and described this syndrome since 1965. Whether there is not cure for this disorder, some symptoms can be treated with physical therapy, occupational therapy, speech and language intervention can help children. The condition of these children is permanent, but the children can still look forward to a normal life. Well, thank you for listening, and hope everyone learned something new today.